As the title says, it's one slide, 3 minutes, and it's all about postdocs in 180 seconds. So I just want to start with this quote that science isn't finished until it's communicated. So as scientists in the lab, we do a lot of discoveries. We find out new things. We invent new tools. But until we communicate that with our stakeholders, in the society at large, I don't think our job is complete. And that's what this competition is about. And that brings us to this cartoon, which is, can you tell me about my science? And like, that's the reaction typically. Because again, as scientists, sometimes we tend to overcomplicate things. But now, as a society, it's more important than ever to communicate science clearly and candidly to everybody, because science is what moves our society forward.
Recording has started.
So what does this number mean? 1,400. Number of postdocs at the University of Michigan. Bingo. Too bad there's no prize for that, but you're absolutely right. That is the number of postdocs at the University of Michigan. So when you think of science, you think of graduate students, you think of undergraduates, you think of faculty members, but postdocs are a vital force in moving our science forward because they are very advanced and mature in training, and they are here at a university like Michigan to advance themselves even more so they can take that training to the next step of their professional career. Okay? So 1,400 are the number of postdocs across the University of Michigan, and if somebody can guess what this number is, this is the number of postdocs, 600, at U of M Medical School. And that is who we are celebrating today. Why today? Why did we pick today? This week happens to be the National Postdoc Appreciation Week, which is a celebration across the country, across universities, a week-long celebration where we celebrate postdocs. And we are no less at Michigan. We are really proud of all our postdocs here and applaud them for the research that they do, not only in the lab, at the bench, but in mentoring grad students, in mentoring undergraduates, and coming out and talking about science like our students, our postdocs are doing today. So that brings me to who I am. My name is Shobha Subramanian. I am the Director of Curriculum and Educational Initiatives at OGPS. OGPS stands for Office of Graduate and Postdoctoral Studies at the University of Michigan Medical School. Anybody guess what this map is? It's the map of Ann Arbor. And I think of my job here, which is helping our postdocs, graduate students, and postbacks in career profession development while they're here in Ann Arbor, kind of a combination of cell biology while they are in Ann Arbor, finding their path. Each of those lines are the arterial roads in Ann Arbor. And my job begins with making our postdocs and students aware of many careers that will end in a successful job search. But to do so, communication, as you can see, is a very important piece. Okay? And communicating science to different audience, to different employers, to different stakeholders is very important. So as in doing Postdoc 180, our postdoctoral scholars hone communication skills, build their confidence as they move along. They get a chance to showcase their awesome research to a broad audience. They engage with the community, like today, and of course, win prizes. Okay? And the prizes are pretty sweet today. The first prize is $1,000. The second prize is $500. The third is $250. And the audience will get to vote live at the end, and that's $250 also. So there are 4 prizes to be given today, so a lot of excitement. So coming back to Postdoc 180, I first want to thank our preliminary round judges who took all the video submissions we got and carefully looked at them, observed them, made notes, and sent us the scores back. And these judges are all from the medical school: Carrie Ferrario, Peter Fedolino, Dave Jones, Mei Hyung, Gillian Piering, and Swati Yadlapalli. And you can see they're from many different departments across the medical school. So I thank them for their service. And now I will introduce our finale judges who are here today. They're all very busy people. And I want to thank everybody, before I introduce you, for coming here, spending an hour with us, and in helping our postdocs and engaging with us in science communication. And can you please stand when I call out your name? So our first judge is Mary O'Riordan. Mary is also our fearless leader. There's a lot of fear. Mary is Associate Dean in the office that I work in. And she's also a professor of microbiology and immunology, studying bacterial pathogenesis and immunology. Our next judge is Andy Rader. Andy is a founding partner of EMA Partners, LLC. And I'm going to read a little bit about what EMA does, because I was reading it this morning. So EMA assists clients in mergers, acquisitions, and intellectual property, licensing, and sales. And Andy has had a long career in this space, working with middle market clients on numerous transactions in many industries, from life sciences and medical devices, healthcare, IT, and consumer products. So he talks to many stakeholders, and convincing Andy is very important for our postdocs today. So thank you, Andy. Our next judge is Dr. Mike Solomon. Dr. Solomon is the Dean of Rackham Graduate School, also the Vice Provost of Academic Affairs for the entire university. He's a professor of chemical engineering and also a professor in macromolecular science and engineering at the College of Engineering. And his research interests are in the area of complex fluids and soft matter. Did I get that right? And last but not the least, Jenny Wilkening, an award-winning high school science teacher from Pioneer High School of the Ann Arbor Public Schools, is one of our judges, and she was awarded the Exceptional Teacher feature a few years ago, and she is very actively engaged in the community for many different things, including being an awesome swim coach. So thank you to all the judges. I also wanted to take... I want to take a moment to thank everybody in our office, because this is the product of a lot of hard work from many different people in my office. In particular, I want to thank Catherine Garber, who's sitting in the middle there. She is our communication specialist extraordinaire, who deals with a lot of different things for communication and marketing. Catherine Melvin, who's in the back there with the camera, she keeps us all together, and we go to her for everything. Aaron Reifler, who's in the back there, is our postdoc studies coordinator, and he has been deeply engaged in this project. And Maggie Evans, who's not here, she is our career professional development coordinator, and she also was deeply engaged in this work. So thank you to all our staff who worked so hard for this event. So that brings us to the real thing, the competition. Is everybody ready? Okay. So, oops. My slides didn't get transferred because I moved from my computer to the desktop. I had put in the rubric that the judges have in front of them. Mary, can I just quickly borrow yours for a second? Sorry. So the postdocs are going to be judged on 3 main criteria today: comprehension, engagement, and communication. Okay? So we want to see if they had enough background and if their talk was in a logical flow. We want to see if they captured the audience's attention and they were enthusiastic, and did they want the audience to know more about their science and their research. And was their presentation clear and concise? and if the content and delivery was appropriate for a lay but educated audience. So that is the rubric that the judges have in front of them, and they're going to be giving points for each of those items. What I will be doing is I will introduce every postdoc who is a finalist today in alphabetical order, and I invite them to come on stage once I read their name, their title, and their affiliation. And the clock will start when the postdoc says the first word. And my iPhone's going to be my timer. And I'm going to wave at you guys at 15 seconds remaining. And then at 3 minutes, I'm going to stand up and start clapping very loudly. That means if you are in mid-sentence, you just have to stop. And I told the judges it's up to them, it's their discretion, what they want to do for disqualification or take points off if somebody didn't complete on time. So they have the ultimate power, whatever they think is appropriate. So without much further ado, I'm going to pull my timer up. Give me a moment. Okay. So our first speaker is Dasia Torres from internal medicine. She's in the lab of Jason Spence, and the title of her talk is Racial Disparities in Esophageal Cancer.
Do we have a pointer?
Sorry?
A pointer?
Pointer. Is it? Yeah.
Okay.
Okay.
Cool. All right. Thank you, everybody, for being here. So, like she said, the title of my talk is Racial Disparities. So there's actually a saying out there that an apple a day keeps the doctor away. Well, my research is actually interested in this question, but in the context of cancer, specifically the cancer that happens in the junction of your esophagus and your stomach. And what we know is that the main risk factors for this cancer is constant acid reflux that damages your esophagus. And what the field has known for the longest time is that this cancer is more frequent and highly prevalent in the Caucasian population when compared to the African American population. Although both populations are exposed to the same risk factor, the reflux, at the same rate. So now, my research was the first one to identify an enzyme known as GSTT2, which actually functions to protect your esophagus against DNA damage. So it protects your cells against damage. And what we found is that this enzyme is actually more abundant in the esophagus of African Americans when compared to Caucasians. Now, my current work is actually to create a large biobank of esophagus tissue that we can actually grow and maintain in vitro. And we can grow it for long periods of time. And what is most exciting about this enzyme is that it's actually inducible. And you can induce it using apple polyphenols, so the phenols on the apple skin. So now we're trying to combine this idea and this induction of apple polyphenols and treat our biobank of Caucasians and African American tissue and actually see whether this can protect your cells against DNA damage and reflux. And maybe we'll be able to answer whether an apple a day will keep this cancer away. Thank you.
Everybody gets a gift. Our next speaker is Kaitlin Hull, who is from microbiology and immunology, and she has two advisors, Denise Kirshner and Jennifer Linderman. And her topic is M. tuberculosis, using mathematical models to better understand disease progression.
tuberculosis. Does anyone even get that anymore? That's how many people react when they learn that scientists are still studying how the immune system responds to infection with the bacterium M. tuberculosis, abbreviated as MTB. However, the reality is that tuberculosis remains a widespread and deadly infection. 1/3, approximately, of the world's population is infected with MTB. That's about 2 billion people. And of those people infected, about 1 in 10 have an active infection, meaning that they're showing symptoms of the disease, such as coughing. Current antibiotic treatment regimens are long and complex, can have severe side effects, and due to the increasing prevalence of multi-drug resistant strains, sometimes don't even work. MTB is characterized by the formation of lung granulomas, which are collections of immune cells that form in response to infection or inflammation and surround the bacteria, as you can see on the slide here. Granuloma formation can successfully contain MTB, but it can also prevent it from being fully eradicated, which means that a seemingly controlled infection can become active later in life. Our lab uses mathematical models to study this immune response. Our model simulates the movement and behavior of thousands of cells and bacteria within the lung environment, enabling us to study the dynamic formation of a granuloma over time. Experimental work is difficult, time-consuming, and expensive, and thus modeling provides a valuable alternative. My work has been to incorporate neutrophils, which are a specific type of immune cell, into my lab's mathematical model. Little is known about the role of neutrophils in MTB. Are they beneficial or detrimental to the host? Does the answer depend on the stage of infection? Although neutrophils engulf bacteria, at least in the case of MTB, they seem to be pretty bad at then killing and eliminating that bacteria. Thus, neutrophils can sometimes be thought of as a Trojan horse. Perhaps they're providing the invading bacteria with a safe place to temporarily hide, away from other types of cells that are more deadly. Through the use of mathematical models in comparison with experimental data, we hope to learn how neutrophils influence the formation of a granuloma, and whether neutrophils might be useful targets for new and more effective therapies. Thank you.
Our next speaker is Jennifer Judge. She is from Molecular Integrated Physiology Department, and her PI, her advisor, is Susan Brooks. And the title of her talk is Live and Let Die, Cellular Senescence Drives Aging.
Just over 100 years ago, in the year 1900, people in the United States were only expected to live about 47 years. In contrast, Americans today can expect to live to almost 80 years. But this isn't as good as it sounds. While lifespan has increased dramatically, we are now a population riddled with chronic disease, many without effective treatments, that can amount to billions of dollars spent in associated healthcare costs. So while it's wonderful that people are living longer nowadays, if those years aren't healthier, then we still have a lot of work to do. And that's what I'm working on. I'm studying how a process called cellular senescence contributes to health and aging. During senescence, cells throughout the body become exhausted, meaning they lose the ability to divide and make other new healthy cells. And they can no longer carry out their proper cellular functions. You can think of these cells like getting a flat tire on a car. The flat is stuck on the wheel, and it's no longer helping move the car forward. It's no longer functional. And depending on where in the body senescent cells accumulate, different organ functions can be compromised and potentially lead to disease. For example, cellular senescence has been shown to be associated with eye diseases, heart disease, arthritis, and even cancer. Senescent cells can also act as amplifiers by secreting factors into their microenvironment that can negatively affect other neighboring healthy cells. But probably most importantly, our stem cells become senescent as we age, meaning they lose that precious self-renewal capabilities. So what am I doing to help combat cellular senescence? I'm wondering whether aerobic exercise can help decrease cellular senescence and be a clinically relevant intervention. We already know that if we prevent senescent cells from ever accumulating using genetic mouse models, the mice will live longer and have less spontaneous disease and incidence of cancer. This is strong evidence that targeting cellular senescence might help us live healthier lives. And I'm beginning to see in my animal models that exercise on a treadmill can decrease expression of many genetic pathways that can cause cellular senescence. And I'm excited to further study specific mechanisms of how exercise might be doing that. And we hope that by using our animal models, we'll be able to gain new knowledge about aging and be able to translate that into potential human interventions. By the year 2050, we're expected to live to almost 90 years old. And I'm confident that by targeting cellular senescence, we'll be able to make those years healthier. Thank you.
Thank you, Kaitlin. Our next speaker is Nidhi from the Biological Chemistry Department, and her advisor is Kaushik Raghunathan. The title of her talk is Unraveling the Adaptive Ability of a Cell.
When I say survival of the fittest, the first thing that comes to mind is the ability to fight back. But have you ever wondered how the basic unit of life, a cell, survives the challenges posed by new and stressful environments? The prevailing notion is that under such circumstances, the only recourse is Darwinian evolution, according to which a random change in a gene might enable its survival. However, such genetic changes are slow, infrequent, they occur over a course of millennia, and are irreversible. But is there a way that presents a distinctive opportunity for how cells might transiently adapt to a new environment? In other words, what changes would occur inside the cell within the immediate time span in response to stress that would enable its quick adaptation? These are some questions that I seek an answer for. So when I moved here just 4 months back for my postdoc, I came across this very recently published article which shed some light on the possible mechanism behind this transient adaptation. And the needle points to epigenetics. Epigenetics is the playful combinations of modifications that controls whether a gene should express or not. That means whether it should make protein or not. Hence, epigenetics is a tool that lets the cell decide which genes to express in what conditions that would best suit the need of that hour. But how do you study these game-changing modifications inside the cell? To answer this, I will use a very powerful and interesting device called a microfluidic device, wherein you can observe the changes occurring inside the cell in real time. In microfluidic device, you let the cells grow in thin capillary-like tubes, and you observe the changes in response to stress. What these changes are, when exactly do they appear, and how they are inherited over multiple generations, giving us an opportunity to look into the decisions made by the cell at every step of its adaptation. I intend to study the factors that contribute to the cellular adaptation process and to find out if epigenetics flexibility is a secret superpower for adaptation. I'm excited to embark on this journey and to pursue my postdoc tenure here so that I can unravel the factors to contribute to adaptive ability of a cell. Thank you.
I'm going to give the judges maybe a couple of seconds. I felt like I've been rushing through. Is it okay? Okay, all right. Okay. Our next speaker is Brandon Patterson. He is from radiology, from the lab of Douglas Miller, and the title of his talk is Investigating the Mysteries of Lung Ultrasound.
Have you ever had an ultrasound or known someone who's had an ultrasound? In medicine, ultrasound is one of the most powerful tools we have for looking inside the human body. Chances are, when your parents first saw you, they probably saw you through ultrasound. And if you have kids, you probably first saw them through ultrasound. We use ultrasound all the time, and it's typically considered to be the safest form of medical imaging we have. However, there is one known side effect, and that's that when diagnostic ultrasound is used to image the lungs of mammals, we've shown in animals that it can actually cause the lungs to bleed or hemorrhage a little bit, and we still don't know why. Now, this is a problem that we've seen in everything from rats and mice all the way up to pigs and monkeys, even though you can't safely test for this in humans. Behind me is an image taken by a colleague in my lab of a rat's lung after it was exposed to diagnostic ultrasound. You can clearly see the hemorrhage. This is a problem that occurs well within the safe legal limits and guidelines for diagnostic ultrasound. And part of the reason for that is that we set those limits before we really understood this problem or before we could even use ultrasound to image the lungs. If we're ever going to set safer guidelines for the future, we need to know what's going on here. We need to understand this problem. For my postdoctoral research to address this, I'm trying to figure out exactly what's physically happening that allows ultrasound to hemorrhage the lungs. Now, there's a lot we still don't know here, but one thing we do know is that that fragile barrier between blood and air in the lungs is breaking down. So to study this, I'm firing ultrasound up through pools of blood at air surfaces like those in the lungs, and using a high-speed camera to see what happens at those surfaces. And I'll admit, for most kinds of diagnostic ultrasound, I don't really see much that's that interesting. Maybe some ripples on the surface, but not much is going on. But for certain new types of diagnostic ultrasound that we use to characterize tissue, what I see are these bloody fountains spraying into the air. Behind me is an image of a similar fountain created in water by firing ultrasound up through a pool of water and exciting the surface. Now, this fountain behind me is only 1 centimeter tall, but it's able to fire drops almost 2 feet into the air, which means that there's a lot of energy here and potential for damage. This is something that we need to understand. And while my research shows that this probably isn't the only mechanism causing diagnostic ultrasound-induced lung hemorrhage, because it just doesn't happen for all types of ultrasound, my research has also shown that this is the kind of thing that we really need to keep in mind and be concerned about when we're setting our safety guidelines in the future. Because of my work and the work that we do in my lab, we're moving towards a safer, better future for diagnostic ultrasound of the lung. Thank you.
Our next speaker is Kayla Peck. Kayla is from the lab of Adam Loring, who is in the Department of Internal Medicine, Infectious Disease. And the title of her talk is Virus Evolution, How Many Typos Should You Make?
Oops, sorry. Oops, that's not me. Yeah. Viruses evolve extremely fast. Influenza virus, which causes the flu, can evolve to look differently in just 1 year. This rapid evolution is one of the reasons we need the flu shot every year. The virus has changed so much that our immune system is unable to recognize it. So what determines how quickly viruses evolve? One of the most important things that influences how fast a virus evolves is its mutation rate, or the rate at which mistakes are made when a virus makes more copies of itself. Mutations are like typos. When a virus makes a copy of its genetic code, a typo can occur such that the wrong letter is added. Some viruses, like influenza virus, make a lot of typos, and this helps explain how they can look different in just 1 year. Most mutations, like most typos, are bad. While a typo can cause a word to lose its meaning, a mutation can cause a virus to not function properly or even die. So if most mutations are bad, why would viruses make so many of them? Imagine you're a scribe, and your job is to copy books by typing them out. You get paid based on how many books you've copied at the end of the day, but your pay gets cut based on how many typos each book has. With these rules, you can take two approaches to your job. One, you could type incredibly slowly, but accurately, such that the books you copy have 0 typos and your pay doesn't get cut. Two, you can type extremely fast and type as many copies as you can, and even if they have typos, you'll make enough money to make up for it. So which of these strategies would make the most money at the end of the day? For viruses, a virus that makes more copies of itself does better than other viruses. But if those copies have too many mutations, then it does worse than a virus that is making fewer copies. So what is the best solution? My research aims to answer this question. To find the best solution, I am making thousands of mutants of a model virus to identify a set with a wide variety of mutation rates. Imagine sitting thousands of people down to take a typing test and selecting a set with a variety of different typing speeds and accuracies. I will give my set of mutant viruses the job of scribe and determine which one makes the most money at the end of the day. Using this method, I can identify the best mutation rate and see whether viruses in the wild are at this rate. Using the book analogy, we can begin to understand why viruses might be making lots of typos. But why do we care if viruses in the wild have found the best mutation rate? Recall that mutation rates determine evolution rates, which have important consequences for public health. Think about getting that flu shot every year. If we can identify the best mutation rate and see whether it matches the rate of viruses in the wild, we can better understand how viruses might evolve in the future. This understanding would allow us to develop better drugs and vaccines. And who knows, maybe one day we'll only be getting that flu shot every other year. Thank you.
So we have 4 more to go. Our next speaker is Dan Schill from the Cell and Developmental Biology Department. His PI is Sue O'Shea, and the title of his talk is Bipolar Disorder: How Are Bipolar Patients' Brain Cells Different? Forgetting to do that.
The World Health Organization estimates that 60 million people suffer from bipolar disorder, and that mental and behavioral disorders are among the leading causes of disability worldwide. This finding struck me, and in combination with other neurological disorders that run in my family, I became interested in pursuing research in this field. Bipolar disorder is a disease that's characterized as changes in mood state, ranging from manic or hyper episodes to sad or depressed episodes. We think that changes in brain cells and how they communicate with one another in both development and in adulthood contribute to progression of this disease. Importantly, bipolar brain cells have been shown to function differently than those that are in undiagnosed people. And my work focuses on better understanding what causes these differences. But wait, we can't just take brain cells from living people and study them, can we? How we're able to get around this problem is we take skin cells from bipolar patients and put factors onto them to turn them into stem cells. These stem cells can become any type of cell in the body, and by putting different factors onto the stem cells, we can make brain cells to study in the lab. One type of brain cell is a neuron, and neurons communicate with one another through electrical signals, acting like a giant circuit board. One molecule that is involved in this process is calcium. And neurons have calcium channels, which help these cells take up calcium to perform this function. Importantly, levels of one of these calcium channels has been shown to be different in some bipolar patients. We are using a gene editing approach to repair this dysfunction that we see in bipolar brain cells and are investigating if it fixes some of the differences we see in bipolar brain cell function. Another type of cell in the brain is astrocytes, and these cells act as support cells for our neurons. They release particles and factors which our neurons can take up. Now, these particles can contain good things that are healthy for our neurons, but they can also contain garbage that is unhealthy for our neurons. We have found that there is differences in these particles in bipolar brain cells and are investigating how these differences impact the cells which they are transferred to. We think that these particles aren't as helpful in bipolar brain cells. In sum, these approaches will allow us to better understand differences in bipolar brain cells and identify new therapeutic strategies to treat this prevalent disease. Thank you.
Our next speaker is Becky Show. Becky is from the lab of Ormond McDougall in the physiology department also. And her title is, Do These Genes Make My Bones Look Fat?
Did you know that your bones are full of fat? All of ours are. There is a unique type of fat cell located inside the bone as part of your bone marrow. In fact, this fat can make up to 10% of your total fat mass. Scientists have known about bone marrow fat for more than a century. Over 100 years later, and we still don't know what it does. One thing we do know is that it's likely more than just a space filler inside the bone, and it may play an important role in many different metabolic diseases. For example, bone marrow fat expands during times of starvation, such as in anorexia. Interestingly, this fat also expands during times of excess energy, such as in obesity. It's weird that bone marrow fat would respond similarly to two very opposite situations. What causes this expansion, and is it important for how our body copes with what nutrients are available? In order to understand bone marrow fat expansion, we plan to feed mice a diet that is either very high in calories or very low in calories, and allow for their bone marrow fat to expand. Next, we'll use a gene sequencing approach to determine how these cells respond to different dietary changes. If we see that certain genes are more abundant in the fat cells of mice that have been fed the high-calorie diet over those on the low-calorie diet, it may suggest that those genes are important for how the tissue responds to excess energy. Now, both obesity and anorexia are times of great stress on the body. And one way the body copes with this stress is with a very important protein, the glucocorticoid receptor. This protein senses stress hormones in the body, such as cortisol, and signals to the cell for it to change its functions in order to survive. We believe that the glucocorticoid receptor may play a role in the expansion of bone marrow fat during times of stress. Therefore, we have designed a genetically altered mouse model to selectively eliminate the glucocorticoid receptor from bone marrow fat cells. We believe that in the absence of this protein, we'll see changes to bone marrow fat cells during expansion. However, even if we don't see changes, the nice thing about our gene sequencing approach is we can identify new proteins that may play a role in bone marrow fat expansion, and hopefully answer the question, do these genes make my bones look fat?
Our next speaker is Charlotte Banneker from Sue Mentor's lab in physiology. And the title of her talk is The Fertility Orchestra, Who is the Conductor?
One of our main interests in life is to have sex. Sex allows us to reproduce. Nowadays, having kids may not be an easy thing, as it is estimated that 1 in 6 women have fertility problems and 20% couples have difficulties conceiving. The reproductive axis consists of the brain, the pituitary gland, and the gonads, which communicate to control fertility. It indeed starts in your brains. The production of neurons in your brain secretes a hormone impulses that are required for the proper function of the reproductive axis. These hormonal pulses suggest coordination of the neurons. Like an orchestra, the neurons should be coordinated to play the proper symphony of reproduction. To do so, they listen to the signals given by the conductor. We think if the conductor is not orchestrating the rhythm properly, this may cause infertility. But in our system, the conductor is not known. Yes, we are in 2018, and we don't know who's driving the hormonal pulses crucial for our fertility. Many scientists think this conductor may be other neurons, but neurons are in fact not the major cell type in our brains. The main cell type in our brains are glial cells, or glia. What are glia? These are cells that maintain the brain together like glue, and were thought to support neurons. But glia is known now to actively participate in neuronal communication. Each glial cell contacts hundreds of neurons, and they are connected to each other. They could thus coordinate numerous neurons. What if the conductor of the fertility orchestra was glia? During my postdoc, I'm trying to understand the role of glia in fertility, and thus its effect on my neurons of interest. Instead of playing music, neurons are electrically active. I'm a neuroscientist, and I'm measuring this electrical activity from mice. We first asked if activating glia could modulate the activity of my neurons of interest. And indeed, when glia were stimulated, we could measure an increase in the activity of these neurons. I also showed that this is due to molecules secreted by glia that stimulate neurons, suggesting a direct communication between glia and neurons. My studies suggest glia could take the place of the conductor of the fertility orchestra. It would support that these cells are far from being passive elements of our brain and could give us new targets to treat reproductive disorders. Next time you're picking a song to set the mood, think about your glial cells. They may be a main actor of your fertility. Thank you.
So our last act today is by Warren Hagen from the pathology department in the lab of Mike Bachman. And the title of his talk is Making Superbugs Less Super.
Finding Nemo, a pair of blue jeans, some old guy, and the poop emoji. If I were you, I would be wondering what on earth these have to do with superbugs. Now, most of you have probably heard of superbugs, either on the news or from that one weird uncle on Facebook that only types in all caps, but we can all say that superbugs are super scary. They're so scary because we can't kill them with antibiotics. They're resistant. And as they become more resistant, we become less able to control them. So, there is some good news for humanity in all this. Only a fraction of superbugs actually cause infection, and the people that get these infections tend to have the superbugs living inside them. So what we do is we root through patient samples, which is as gross as it sounds, looking for the superbug known as Klebsiella pneumoniae. One could say that we spend our time finding pneumo. And when we find these guys, we dig through their little bacterial genomes looking for genes that help them cause infection. So you may be wondering, why spend time and money doing this when we could just make new drugs? Well, it turns out that making new drugs is extremely expensive, it takes a really long time, and it's very challenging. So we hope that by understanding how superbugs cause infection, we can come up with new and creative treatments. So this research has already led us to a gene discovered by this good-looking fella. Now, for those of you that aren't microbiology buffs, this is Sir Alexander Fleming, who's very famous for his very accidental discovery of a compound that he lovingly called mold juice. Today we call it penicillin. Now, I'm not here to wax poetic about Fleming or penicillin, but I do want to talk to you about a different chemical that he worked on known as tellurite. Now, if you're my dad, you make a bad joke about the Telluride Film Festival. It's got nothing to do with that. Tellurite is a compound that's toxic to humans and bacteria, and it turns out that our favorite superbug, Klebsiella, has a gene that resists tellurite. And when this gene is present, a patient is up to 150 times more likely to get an infection. So how does this knowledge actually help us fight superbugs? Well, that is where the poop comes in. Now, poop is amazing stuff, and it's really easy to get a hold of. So if we can find tellurite resistance genes inside patient fecal samples, we can diagnose a patient's risk of an adverse outcome. Put more simply, a quick butt swab when you get to the hospital, and we can tell you if you're likely to get an infection. And if you are, rather than wait for you to get a disease that we can't treat, we can put you on simple and cheap precautionary measures to prevent that infection from occurring in the first place. So by diagnosing infections before they occur, we hope to make superbugs a little less super. Thanks. Thanks, Shoba.
So that was an amazing set of talks. So let's give a round of applause to all our speakers. So like I promised, the audience will vote. Hang in for 5 minutes while I take the judges and tell them their drill. So think about your thoughts and pick your top choice because you only get to vote for one person. So the poll will go live in about 2 or 3 minutes, so just hang tight. And when it goes live, I'll put the screen up, and then you can pull out your phones and either text it. It's like, what's the show? America's Top Model or whatever, something like that. You can text your results, or you can vote in. There are many different options to do it. So, but just think about it and pick your top. laser pointer. Okay, audience. So here is the prompt. So you can go to pollev.com/pibs, and the poll should be live. And you just click on one of the 10 choices as your vote. Or you can text and then type the letter. It might be easier to do the pollev.com/pibs. So Catherine tells me it's live now. So cast your vote. Looks like most people are done voting. Anybody still voting, raise your hand, and we'll keep it live for another moment or so. A couple people still are, okay. All right, I think safe to close the poll now. All right, thank you. So I want to make a quick announcement. So Jay, who was our last presenter, is also the president of the Postdoc Association, along with Francois, who's sitting in the, Francois Buffet, who's sitting in the back there. And I'm told that all the presenters will get a free registration to present their 3-minute talk at the Southwest Regional Michigan Postdoc Symposium. So congratulations. So the judges should be coming back with their results, which will be announced. And after that, there's a small reception outside, so feel free to mingle with all the postdocs, congratulate them, celebrate with them, and if the postdocs can just take a picture at the end together before you disperse. Okay, everybody ready? I'm excited. So, this is like the Academy Awards. Because I don't know, I don't know, I only saw the first one yet. The third prize for $250 goes to Dasia Torres. Second prize for $500 goes to Charlotte Banneker. Congratulations. And the first prize of $1,000 goes to Warren Hagen. Congratulations. And who did the people vote for? Don't sit down, Jay. It's J. Warren Hagen. Thank you, everybody, for coming. Before you leave, I want to invite Mary O'Riordan, our leader in our unit, to say a few closing remarks.
Well, I would like to thank you all for coming, and we can talk about some lessons learned for today, one of which is we have some incredibly talented postdoctoral scholars here, so wow. I think the second lesson learned is put a poop emoji in your talk. I'm doing that from now on, even though I'm not sure it's that relevant to what I'm doing, but that's okay. But also, to realize actually the power of our research and how we communicate with the public, I think is something we can all continue to work on and showcase. It's pretty easy to talk to each other about what we're doing and how exciting our research is, but I think that really distilling what we're doing down to its first principles and practicing talking about what the important problems are that we work on here every day is incredibly important. And part of what we're trying to do in our team is think about ways that we can showcase the work that you all are doing in different ways and trying to really make this more available to the public. So, keep an eye out for more things like this. This competition has been really exciting, and it was fun for all of the judges to watch. We agreed that this was a very, very difficult decision. You guys made it really hard on us. So thanks for all that. But thanks especially to Shobha Subramanian, who I think has just brought so much innovation and thought and passion and organization to really showcasing and helping our postdocs and students make the most of their education here. So thank you all. Please join us for a reception out here. Then we can all sort of chat further and learn a little bit more about what the science that we're doing here. Thanks for coming.